I will focus on the physiology of human space exploration that has been discussed in the previous two lectures and maybe expand a bit. And I guess as a result of Jennifer's talk and of course Justin's, we now see we have to have the force with us, the G-force, as we go into space. So this slide shows basically our facility. It's in my period of my career, this is the only lab with a view. So we like going, this facility that we have is here. That's the Olympic Sports Center. And this is where we conduct most of the bed rest experiments that I will talk about. When discussing the relevance of space life sciences, I always like to give the example or start with the example of James Lind, and it's very appropriate that I do this at this presentation. James Lind was a Scottish physician who worked in Haslar Hospital just across the way in Gosport. And at that time, of course, we were exploring the unknown regions of the Earth, and many of the ship's complement died, but not of any maritime disasters, but something as simple as scurvy. And James Lind is attributed of conducting the first controlled medical experiment where he actually introduced citrus fruit into the diet of some ships and not others. And of course, the ships that had the citrus fruit came back with almost a full complement, therefore showing that this was an important feature. They didn't know about vitamin C at that time. But interestingly, that knowledge, of course, was then used by everybody else, not just the explorers. And this is the same thing when we talk about human space life sciences. It's not just for the handful of astronauts, but it can actually help certain populations. And as a result of his work, the Institute of Naval Medicine, again in Gosport, has in its logo a lemon tree. And that's why. So in the same sense, we're now trying to investigate what are the problems of living in space on the International Space Station. And Jennifer already reviewed basically some of the problems. I will focus on the problems of low and zero gravity. So again, using her slide, but now I would like to categorize the problems that we face in space into two. I'll call them in-flight problems and post-flight problems. In-flight problems are problems where the physiological systems have not yet adapted. And an example of this is space motion sickness, something that occurs within a few days of space flight and then subsides. The in-flight problems are the main problems. These are problems where the physiological systems have adapted. And this is great. I mean, on Earth, as we've been discussing at this meeting, if we go to altitude, heat, cold, and so on, the organism adapts to those environments, and it's beneficial. And when the organism returns to its original habitat, of course, you have de-adaptation. But with spaceflight, when the human body adapts to microgravity, then return could be actually very detrimental to the astronaut. In fact, it could be life-threatening. So the research in space life sciences has gone along two pathways. One is to try to understand the process of adaptation. And the second pathway, as Jennifer said, the countermeasures, is trying to prevent this adaptation from happening so the astronauts can return safely to Earth. So of the post-flight problems, we have the cardiovascular deconditioning, musculoskeletal deconditioning, and as Justin just mentioned, spaceflight-associated neuro-ocular syndrome. So what we need to know is that all the physiological systems do not adapt at the same rate. There are different rates of adaptation, and this slide shows this challenge. So if we start with a sort of, I don't like using the word set point, but for this purpose, let's call it the reference point of 1g, and we then go into space, so to 0g, what we have is a response of the various systems to this change. And what you can see is that in some cases, we actually reach a change that on Earth would be considered a clinical threshold. There would be concern that this is clinically significant. And then after a while, they subside. So the fastest system is the neurovestibular system. And of course, the slowest would be something like the bone demineralization. I've added a red line to this graph, which is to show that these changes in vision also occur after about a few months in space. And what we think now in the majority of the cases, these changes are irreversible. That's why this is almost like a showstopper for future deep space exploration. And as they return to Earth, the same kind of readaptation with the same kinetics occurs. The facilities that were mentioned before, of course, from looking at physiology of space flight. We don't have to go to the moon. We can do a lot of this work here on Earth. So the bed rest facilities obviously exist in the Space Medicine Institute in Toulouse, the Aerospace Medicine Institute in Cologne, and the Olympic Sports Center in Planica. And I will focus mostly on the work that we've been doing in Slovenia. We've also been doing some work in the Concordia Antarctic base. And the interesting thing about Antarctic base is not only that it's isolation that you can look at, but as I'll later in my lecture, I'll show you that now hypobaric hypoxia is also an issue with future space flights. So in fact, you can actually look at hypobaric hypoxia and isolation at the Concordia station. Not only is it 3,200 altitude, but because of the lower absolute pressure on the poles, the actual partial pressure of oxygen in the station is more similar to about 4,000 meters. So it's quite a significant stimulus. And because of this hypoxic issue that has arisen in Planica, Slovenia, we've actually been looking at hypoxic bed rests, and I'll get to that later on. So first of all, bed rest has been mentioned, and of course, is this a good model or not? Well, the bed rest, I think about over 100 years in 1850, Queen Victoria's physician, I think his name was James Hilton, suggested bed rest is good. It can solve a lot of problems. If you're ill, go to bed for a long time. And in the 1940s, I think it was Asher and then Browse who suggested that this is really bad for you. They actually wrote a book, The Pathophysiology of Bed Rest. So it's not a good thing. But there are similarities between the changes that occur during bed rest and the changes that occur in astronauts in space. And that's why this model is used. So microgravity, when you think about it, it unloads the weight-bearing limbs, and it causes inactivity. So these are the two main things that actually occur. You have demineralization of weight-bearing bones, and you have reduced muscle mass, which leads to reduced muscle strength. So if you're going to space, this is data from spaceflight data from NASA, where you can see that for a 6-week spaceflight, you have about a 2% to 5% change in bone mass. So this is looking at the calcaneus bone. And you also have about 10% loss of postural muscle mass and about 25% loss in postural muscle strength. So this is spaceflight data. We did a similar experiment where we did 35 days of bed rest, normoxic, and what you find on the left-hand side are the cross-sectional area of the various muscle groups: gluteal, thigh, knee extensors, calf, and ankle plantar flexors. And what you can see for the knee and calf is that we have about a 10% decrement or loss of muscle mass, which is basically very similar to what you see in space. Also, over a 35-day period, shown on the right-hand side, using peripheral quantitative computer tomography, we've seen changes in bone mass as well, so bone demineralization. And what is at different parts of the tibia and femur. What is interesting from these studies on the right is, in fact, and this is something surprising that we didn't know then, is that if you look at the bone, which comprises of cortical and trabecular bone, you have much more loss of the cortical bone than trabecular. So this is actually was surprising, since obviously the cortical bone is the shaft part that gives support. And then there are cardiovascular changes alluded to by Justin, using Raoul's graph here on the right. Raoul, when in his book, classic book, obviously showed this for upright and supine, but in fact, you can also say that this is what happens in normal gravity and microgravity. So you have a loss of this gravitational vector that acts along the blood vessels. You have this shift of blood volume, which gives the puffiness appearance, and that's why we do bed rest sort of with a 6 degrees head down tilt. You have a change in the baroreflex and also a decrease in plasma volume. And so the deconditioning that we followed in a bed rest is actually you have similar responses as in space. We've also seen myocardial atrophy. We've seen reduced myocardial contractility. And the most interesting finding we saw at that time was actually a change in the distensibility of the peripheral veins and arteries. And this is the graph on the right that shows basically the descending pressure and the percent change. And it's a bit of a crazy experiment, maybe difficult to describe, but on the picture you see someone lying in a hyperbaric chamber with their foot sticking out of a hatch, and we've increased the pressure inside the chamber slightly in graded manner. And as we were doing that, we were measuring the diameter of the blood vessels with an ultrasound to get the graphs. And you can see that the bottom graphs, the blue, blue is before bed rest, the black is recovery after bed rest, but immediately following bed rest, you have the red graph showing this increased distensibility, which we see both in the arteries and the veins. And as a result of this total cardiovascular and muscular deconditioning, obviously we have a decrease in our aerobic performance. And this is why, as Jennifer said, we have a lot of countermeasures which involve exercise on the International Space Station. It's a bona fide gym up there with all the exercise equipment they have. And on the right is Sunita Williams, which is of Slovene origin. And I have her there because she was the first to run a marathon in space. So when the Boston Marathon started, the gun went up, she started running as well and completed the Boston Marathon on the ISS. And now the question is exercise really the most efficient countermeasure or not? And finally, of all these questions that are of concern to us, it's the spaceflight-associated neuro-ocular syndrome. And what has been observed in astronauts that have spent 6 months or more in space is these problems such as optic disc edema, choroidal folds, globe flattening, cotton wool spots, basically resulting in reduced vision. This was not so well described before that. Why? Because before astronauts were younger, now they go up at 50. And so at 50, you start having to wear glasses anyway. So it's not something unusual that you have to start wearing glasses. Also, the time spent in space is more. And there's another important feature that I think is the key: exercise. Astronauts now have a regime of exercise which takes about 2 hours of the day, doing aerobic exercise, doing strength training. And when you think about the exercise pressure response and you add that to Justin's talk, you might actually get a significantly greater pressure than what he actually showed. So now, going further now, we've talked about the space station, but there's this feature in space which should also be mentioned, and this is the activities that were called extravehicular activities when people go out to do some work. They don't occur very often. It's about once a month. So in the last 20 years, there have been about 200 of them. But when they do this, it's all hands on deck. I mean, this is not a simple procedure because you're going from a 1 atmosphere environment to a vacuum. And actually, you're reducing the pressure in the suit to about 1/3 of an atmosphere. So the whole procedure is lengthy. It takes about 4 hours because you want to prevent decompression sickness. So you have to reduce the pressure gradually. You have to pre-breathe oxygen, and this takes a while. And it's 4 hours every time. The question now is, if we're going to visit the moon and Mars, and we're in these lunar modules, does that mean that every time we want to go and have a look at a rock, we're going to have to spend 4 hours actually preparing to go out? This might actually be a daily occurrence, and perhaps we might do this several times per day. So this procedure that's on the ISS is not tenable now for these missions to the moon and Mars. So the idea that has been promoted by engineers at NASA has been to actually have a pressure which is lower in the rover, shown on this side. So in other words, have a pressure which is equivalent to the pressure inside the suit, and to increase the oxygen to about 30%. So in essence, you'll have astronauts living in an environment which is somewhat between 3,000 and 4,000 meters altitude. And this is the idea. So here you have the rover with the 2 suits hanging at the back there. And on the top, you see an astronaut wearing the suit. And the procedure is that they would open a hatch, walk into the suit, close the hatch, and basically pop off the rover. So he's carrying basically the frame of the door on his back, as you can see. That's the current sort of thinking. The point where we came in is, okay, if normal baric hypoxia is potentially the environment in future habitats, we know the adaptive processes that occur in normoxia. We've been doing that for the last several decades. Now, if you include hypoxia, how does hypoxia influence these adaptive processes that we know that we've been studying? And so ESA convened a topical team to discuss this issue. And the consensus was that with the available information, nobody knew, in fact, how hypoxia would affect muscular deconditioning, cardiovascular deconditioning, and so forth. So a series of experiments over the last 6 years or so has been conducted in our facility, where we've looked at this issue of hypoxia and deconditioning. We've done all the experiments from 10 to 21 days on both males and females. And we've looked at, we had subjects participate in 3 campaigns. So they would be basically in hypoxia, but ambulatory. And we compare that with being bedridden in hypoxia. And the third campaign was normoxic bed rest. So these are the 3 campaigns. And what you have is the environmental conditions. And just to maybe outline this, we don't actually force our subjects, as shown here, to be in bed. We've not lost a subject yet during a campaign, but they are told they can leave, so it's not that problematic. These studies that are done, obviously, are done in collaboration with several laboratories. We do a lot of studies, and I will only review some of the critical ones, not all of them because of time. But obviously, we're interested in food, nutrition, anthropometry, aerobic capacity, muscle strength, bone mineral density, and temperature regulation. So basically all the issues that Jennifer has listed in her slide. And it's very important that we have subject compliance, because if you have someone in bed and you close the door and he starts doing push-ups or dancing, then it might kind of destroy your results. So subject compliance is really important in these cases, so we can actually interpret them properly. Looking at the cardiovascular system, first of all, what you find is you have changes, obviously, in the plasma volume, which are during bed rest, which are exacerbated in hypoxia. And you have the anticipatory transient change in EPO. But more importantly, on the left here, you'll see the changes in oxygen, in maximal oxygen uptake or peak oxygen uptake. And essentially, we know from before that you will have a decrement in bed rest. So this is going from pre to post and normoxic bed rest, with hypoxia actually exacerbating. Not only this, but also another interesting thing is that it counteracts the orthostatic intolerance that we see. So the other one is muscle atrophy. Now, in 10-day experiments shown here, we don't actually see any significant influence of hypoxia. But we went to 21 days, then there seems to be a significant separation, a much greater effect of hypoxia on the changes we see in the thigh muscle. So hypoxia seems to exacerbate the loss of muscle mass, in this case in the thigh, something we did not observe in the calf. And we didn't see this in the shorter duration exposure. So going to 60 days might actually then provide some new insights into these changes. And as was mentioned by Justin, the ophthalmological issues are very important. So over the years, we've also looked at this. We've initially just looked at the changes in the arterial and venous vessels in the retina, seeing changes shown here during normoxic and hypoxic bed rest. So we have changes in diameter. So in the following experiments, we've actually done the same thing, ocular coherence tomography, and shown here. So if you look at this slide, it has a green circle there. And basically what you do is, if you can imagine, that's the region that's of interest, and now you take that and you spread it out, and you see all the layers beneath. So what you see here are all the layers of the retina. And because we were doing this in hypoxia, and we know that hypercapnia or elevated CO2 is an issue in the space station, we looked at the effect of normoxic bed rest, hypoxic bed rest, as well as with hypercapnia. And what we find is that the choroidea, as Justin said, shown here, actually is affected by the hydrostatic factor, whereas the retinal layer here is not affected so much by the hydrostatic factor, but by the hypoxia and hypercapnia. So you have changes in this diameter so it's sort of chemosensitive, and this one is more due to the hydrostatic. And we've done more experiments now to identify how exercise and hypercapnia actually affect this even more. And for those of you who go to altitude, of course, and there's many of you here, we all know that there are problems at altitude with sleep. And we've also looked at this using polysomnography. And this slide here shows you, on the right, the results show you the apnea-hypopnea index. So basically, there's a national standard. You monitor breathing during night. And if someone stops breathing for more than 10 seconds, there's certain issues, you claim that is a apnea-hypopnea sort of event. And what you have here is the number of events per hour. So on the first day of hypoxic ambulatory, there could be 20 events per hour, whereas on the 21st day, this increases to almost twofold. So in fact, we don't see adaptation in hypoxia, but actually exacerbation of the problems we have in sleep. And of course, the sleep architecture is disrupted. And the slide on this side shows you the total, this is total sleep time, and it shows the oxygen saturation. So if we're looking at normoxia up here, so baseline normoxic bed rest, you can see that we're basically in the high 90s. So if we go to bed, of course, oxygen saturation decreases slightly, and these are still baselines. But if you go to hypoxia, this is bed rest. In fact, at night, you're in these sort of low 80s in terms of saturation. And in fact, this is obviously then the reason why you have these changes in breathing pattern. So to complete this, as Jennifer mentioned, artificial gravity is now perhaps this magic bullet that might help and cover the whole issue. So the issue has been with us since almost 90 years. This is a book which was published by Herman Nordung of Slovene origin. His name was Potočnik, like our, is that a relation? Anyway, so he suggested in 1929 that the space station, if it ever was built, should be circular and rotating to actually establish a 1G gravitational vector, which would prevent any possible deleterious effects of microgravity. But of course, at that time, they didn't know how microgravity affected the humans. This, in fact, was used then by Arthur C. Clarke and then in Kubrick's film, 2001: Space Odyssey. And as Jennifer said, this is now what we're investigating. This is the ESA short-arm human centrifuge. And the research going on now is to try to find out the strategy of using this. How many times per day or how many times per week, for how long? So should we supplement this with other types of exercise while you're actually being centrifuged? So these are the issues that will be on the topics for the next 5 years. And this, if it works, is sort of the way the centrifuge works. This is in the Space Medicine Center in Toulouse. So this is the type of centrifugation that you would then elicit. So at the end, I'd just like to acknowledge the funding agencies that have supported our work over the years looking at this issue. And of course, I'm actually presenting some of the results of a large group of individuals from all over Europe. And I have to thank them for helping us in this endeavor. And if you're ever in Slovenia, very, very welcome to Planica. Thank you very much.